Okay. All right, let's get started. I have one announcement, well, reminder and announcement. So the project due date is Friday, December 9th. But I was convinced by one of you to move it to Monday, December 11th, 12th? 12th. So you will have an extra weekend. What? Are you thanking God? Thank me. You just did this. It's just too mean. Okay. And you can thank Sarah. She was the student. Yeah. Okay. So yeah, make sure that you do well, work on it really well. You have extra time. And what I wanted to say also is the summary, the preliminary summary that you worked on and I corrected for you and you met hopefully your industry mentor, you should have a final summary. That is due with the project, but I want you to send it to me electronically. So you email it to me rather than give me a hard copy. I want the hard copy of your project report, but an email of the final summary. And that I will compile all your summaries and put them in one document and put them on Moodle. I would like each one of you to look through them to see how different each products are and how different the testing would be for different ingredients. So there will be a question on that in the exam, final exam. So I don't expect that you study the principle of each of these methods and the reason and all of that. I just want you to know that different ingredients require different testing and different products require different testing, at least to have an idea, a global idea of that. Okay. The other thing here on Moodle, I have put up the pH and titratable acidity problem key. I did not have a question in exam 2 on this. This means it's still open for testing in the final. So make sure you know that. And the quiz is on Monday. The quiz on Monday is going to be the less material, only the fat characterization and the protein characterization for the quiz on Monday. And let's see. You have the fat characterization practice problems due today, so hopefully you take advantage of the extra credit and submit it today. There is a protein separation practice problems, and I will post the key after you submit. You can do that next Wednesday, so that it's not due on Monday with the quiz. Okay. You can start peeking at the protein analysis review, it should say not revision, review questions. We will go over these questions in the review session that we will have instead of the lab in the last week of classes. So not next week, the following week, instead of having lab, you'll come to lab, but we'll go over these questions, any questions you have, as a review session for the entire material before the final. So you can start having your peek and see some of these questions might actually show up in your final. So it's important to review. Yep.
So do we meet in the lab or here?
In the lab. So like you have a regular lab, you just, instead of having an experiment, we'll have a review session. Okay, I think these are the couple of reminders I have. Okay. I pulled this out of the protein separation practice list, and I thought it would be good to discuss this in class. And if you take good notes, then you have answers for one of the questions in the practice set. Anyway, what I want you to do is spend a few minutes looking at this question, this is a combination, obviously, of your understanding of chromatography, especially ion exchange chromatography, and an application specific to proteins. So take a few moments, think about it, and then we'll have a discussion. One thing you need to remember or know, what would be the charge of a protein above its isoelectric point? The net charge of a protein above an isoelectric point. Negative. Yes. And below the isoelectric point would be mostly positive. Okay, so with that in mind, think about this question now. If you have an idea, we can start the discussion. You want to give it a go, Joshua?
Sure, yeah. I think this is the PI of the solution. Is about 4.5, so they will carry the positive charge. So on the inside, I will use
Carry the positive charge where? At what pH?
at pH like 4.5.
At pH 4.5, it's isoelectric. It doesn't carry a charge. Which ones will be carrying a positive charge? Do you mean these? Or do you mean this? The protein that has an isoelectric point 4.5?
I mean the protein on
At the isoelectric point, you don't have charge. You have a net zero charge.
I would use cation exchange because since if you use a buffer at 4.5, then that means your proteins that PIs at 6 are going to be positively charged. So you want them to interact with the column.
But that will precipitate out.
Oh, so then it's on the column.
You
So if the
don't wanna-
protein precipitates, does it just sit on the column?
It will precipitate, yeah, it won't. Or it won't even reach the column, it will precipitate.
Okay.
If you have a buffer with 4.5, your protein will not be in solution to ingest.
We should have it below 4.5, say 4, so that it has some negative charge.
What will have negative charge?
No, both will have positive charge. We should have it.
It is confusing, I know. Jenny, you want to give it a go?
Yeah, okay. You would use a buffer that's between 4.5 and 6. So the protein.
Golden buzzer. Do we have a golden buzzer?
So at that point, the protein you want would be negative, and the other ones would be positive. So then you'd use anion exchange.
Yes. Bingo. That is true. We don't care about these proteins. Okay? We care about that one protein. So if we go above the isoelectric point and below this, so say 5 or 5.5, this protein will be negative, these will be positive. And if we use anion exchange, that means you're exchanging anions. This protein that you want will interact with the column. The positively charged one will go with the wash. Okay? So which protein will elute in the wash or proteins? If we use that strategy, the proteins that you don't care for will go with the wash because positively charged, they won't interact. Very good. Any clarification? Yes, Jan.
So if we just set the pH at 4.5 and then the bond with 1 will just participate, right? Is that?
Yeah, but we want to use ion exchange chromatography. Yeah, that's a smart point, which I'm going to ignore. Yeah?
The reside the buffer at the pH 4.5, both proteins will the precipitate the come out together. Then how can we separate them?
At pH 4.5, this will precipitate. These will remain in solution.
Okay, so they will not come out together.
They will not come out together, but if you use a buffer at pH 4.5, your protein is no longer in solution, cannot inject it in a homogeneous way onto your column. So you don't want to use a buffer at an isoelectric point of any protein. Yeah. Okay, so that's settled. How will you elute the protein that binds to the column? And why would this work? So you have now gotten rid of the positively charged ones, and then you have that one that you want to retain. How would you expedite it leaving the column? Now what do we do? There are two ways that you can do that. Sven? Huh? Deprotonate the column. Oh, so the column is positively charged. So you want to do something to shield those charges. So how do you do that? Yeah, how do I add negative charge to the column? What do I add to my mobile phase? Yes, Farah? Salt. So if we add salt, then we shield those charges. Ben? There's another method.
Change the pH?
Change the pH. In which direction? So I don't want my protein to interact anymore. It is negatively charged, and I don't want it to interact anymore. Lower the pH. So if I lower the pH to say 3.5 or 4, then my protein will start carrying a positive charge and will lose interaction. So two ways, either increase the salt or decrease the pH. Or both sometimes. If you have two different proteins that are very close, one is at 4.2 and one is 4.5, and you want to separate both of them, then you'll do gradient elution. And then, Yanji, if you have two proteins very close in the isoelectric point, then we need to use ion exchange chromatography. I will change that question for next year. All right. Well, there you go. That's all right. So moving on. Last time we talked about how we can separate protein based on solubility and absorption characteristics and denaturation, and now we're moving on how to separate them by size. So let's say you have a precipitated protein using salt solution, okay? And you want to remove that salt. You don't want to keep that protein living with the salt because it's impacted its functionality. So you want to get rid of the salt. What most common way of getting rid of the salt is something called dialysis. And then Chelsea prepared this demonstration for you because it's always better to see 4-dimension. Is it 4-dimension? Or no, 3-dimension. I'm going to make it 4. Yeah, I'm going to throw it at you in a minute. So it becomes 4-dimension. So this is basically a tube with pores. And I just ruined the assembly. But anyway, so it has pores, and the pores they have specific size. So you can order membranes, they call them membranes, with different pore sizes. So you can say, okay, I want to make sure I retain all my proteins that are even peptides as low as 1 kilodalton. So I make sure I purchase membranes that have 1 kilodalton cutoff. Or I don't care about anything lower than 3.5 kilodaltons. So I get a membrane with pore size that allow 3.5 kilodalton. And then I put my solution with my protein and salt. Now this is a blue dextrin solution just to see it nicely. But usually your protein solution is not blue, it's colorless. And it has lots of salt. So you put it in water. So here's my 4D. You can pass it around. So I put it in water or buffer. And then what happens by osmosis, your salt will start moving out of your membrane into the water. And when you do that, you keep it in the water or buffer for a few hours, and then at some point you reach equilibrium. The amount of salt outside equals the amount of salt inside. So you still have salt in your sample, so what do? Logically, change the water. So I throw away that water, not the tube, the water, and put fresh water or fresh buffer, and continue that for another equilibrium. And I do that at least 3-4 times, until the concentration of salt that remains inside the tube is very, very negligible, very, very small. You are always at equilibrium in this procedure, so you want to reach a very, very low salt concentration. Oftentimes, the membrane, the water goes in too. Salt comes out and water goes in as well. So what happens, your membrane will bulge. And if you have a lot of sample in there, the membrane will burst. Claire's laughing because it happened. True life story. So what you usually do is either put less solution in your membrane and more length of the membrane, or what you can do is make sure you have a membrane that is has a layer of polyethylene glycol layer, and that kind of prevent water permeation. Basically, it concentrates the sample for you. Okay. Another common procedure, and this is not necessarily for desalting, it is mostly to separate proteins of different sizes. So let's say you have a mixture of proteins and you want to get the high molecular weight proteins and get rid of the low molecular weight proteins, or vice versa. Get rid of the high molecular weight proteins and any high molecular weight substances or macromolecules you don't want, and retain the low molecular weight protein. So you're either interested in the permeate or retentate. So it depends on the experiment. So in this case, these are very commonly used. They're called, well, the company that I know it by is Centricon, but there are different companies with different names. But what you have here, and there are different styles of these tubes. There are the small ones that you see in this picture, and there are different types here. So basically, these you can centrifuge. They can put in the centrifuge. There's a membrane in there. You put your solution on one side, you spin, and then by centrifugal force, you get permeate go through and retentate remain. And oftentimes you do a couple of washes to make sure that you get all of what is less than the cutoff. So the cutoff can range. This is a 30 kilodalton cutoff. There is a cutoff of 1, 2, 3.5, 4, whatever. There are a lot of different cutoffs in the market depending on your research. So you can pass around. So depending on the size of the membrane, you can have either microfiltration, ultrafiltration, or nanofiltration, or reverse osmosis. So in industry, there are membranes on large scale, and these are used to isolate proteins from different matrices, for example. So this is the range for microfiltration, ultrafiltration, nanofiltration, and reverse osmosis. So anything greater than 500,000 kilodalton, so 500,000 500,000 daltons or 500 kilodalton. Anything greater than that can be retentate in a microfiltration. And then ultrafiltration has a range. It goes from 1,000 to 100,000 daltons. And then nanofiltration is very small, so you can have a much smaller retentate, a molecular weight of the retentate. And reverse osmosis is mostly for ions, for salts, to remove salts. So here's an example of, let's say you have a milk system. So if you're using microfiltration, you will be removing bacterial cells, and you're going to be removing fat as well. However, protein, lactose, minerals, water will pass through. Get to the ultrafiltration, protein will be retained. So that would be your retentate in ultrafiltration. But then also within ultrafiltration, you have different membrane sizes. So you can select the membrane size, pore size that you want. And then lactose, minerals, and water will pass. In nanofiltration, you can, your lactose, you can get the lactose out. Whereas the minerals will pass through and the water will pass through. So if you want to remove minerals as well, there's the reverse osmosis membrane. So yeah, that's a demonstration of the different levels of membranes that you can have and what would be a retentate, what would be a permeate. Of course, there's chromatography. There are a lot of applications of size exclusion chromatography in the protein world where you separate based on size. For example, one common thing we do in the lab, our lab, is looking at gluten proteins, and they vary in molecular weight size, in the size of the polymers. So what we do in order to determine the different protein ratios, let's say gliadin to glutenin ratio, we run size exclusion chromatography and separate them based on size and determine a ratio that way. Also, it's very applicable if you have a process that resulted in aggregation or polymerization of your protein. So you want to see the level of polymerization. Did you end up having dimers, trimers, large macromolecules? Then you can run size exclusion to see the effect of processing on your protein size. So it's a very, very useful tool, especially in protein research. And oftentimes it's linked to multiple different types of detectors to aid in the quantification and molecular weight identification. Okay, yeah.
Gel filtrations, I just want to understand what you said. When the proteins have aggregated or polymerized, the gel filtration can help you sort out.
Yeah, because
different proteins have gone with the aggregate?
Yeah, because you can have a dimer, and then let's say if you have a protein that's 14 kilodalton originally as a single protein, and then you did something and then it interacted via disulfide interchange, and then it becomes a dimer, then you will get a 28 kilodalton. Then you know, oh, okay, so this particular protein became a dimer. And then if you heat more, have a more harsher process, you might end up with 100 kilodalton protein. So you will have a chromatogram showing you a native protein, the molecular weight of native protein, and the molecular weight of a protein that went through processing. And you can see what kind of polymerization happened, or the level of polymerization. Okay? And then if you remember, the smallest molecular weight will be retained the most, because it will go through all your pores, whereas the large molecular weight will go through less pores and will elute faster. So larger ones will come out first and smaller ones will come out later. Okay. Other questions? That's my favorite, electrophoresis. These are actually my hands when I was a postdoc. Just saying. Took pictures of them. All right, so basically, electrophoresis, we use it to separate protein for qualitative determination of the range of different proteins you have in your sample. And it's not a quantitative measure usually. You can do quantification if you take the intensity of the bands and do a densitometry, then you can have and run standards on the gel, then you can have semi-quantitative results. But oftentimes it's just used to observe what kind of proteins you have, what's the molecular weight range of the protein you have. Again, you have polymerization. If you see bands at the top of your gel, that means something happened to your sample that the proteins polymerize and no longer are small enough to go through the gel. So to back up a little bit, most common electrophoresis used is SDS, polyacrylamide gel electrophoresis. So this is an abbreviation. Sodium uracil sulfate is a reagent that we mix with a sample. So you have a protein solution, you add to it, we call it a Lemley buffer, and in that Lemley buffer you have SDS. The SDS is going to denature the protein. So it's going to break non-covalent bonds, whether hydrophobic hydrogen bonding or ionic bonding. It will break them so the protein will denature. And in addition to that, it will interact with these charges on the protein, and it has a negative charge. So you end up having all your proteins having a negative charge on the surface. With that, your gel that you can make gel, or you can order gels, have made up of acrylamide. So they will have, depending on the concentration of the acrylamide, they will have either small pores or large pores. Sometimes you can have a gradient that vary in pore size. So when your proteins all have the same charge and you run a current, what happens is all of the proteins are going to move in one direction when you run that current. So all of them are going to move down and will be separated based on size. So the smallest will go down the furthest and the largest will slow down. So we won't be doing this in the lab in food analysis, but you will be doing it in chemical reactions if you take chemical reactions. So we will run protein samples to look at them. So basically I have a gel here to demonstrate. Since you're not going to do it in the lab, I brought some demonstrations with me. So in the olden days, when I was a student, very, very long time ago, sort of very long time ago, they didn't have pre-made gels that you can buy. So students nowadays are very lucky. In the past, we used to prepare our own gels even from powder acrylamide. So what a wonderful thing to do, right? Because now you can tell Pam has something in her mind because, you know, it affects the brain, the acrylamide. So I prepared a lot of those in the past. So basically, you would prepare your own gels, but nowadays you would get a cassette. It's actually two plastic sheets, and inside of them they have a acrylamide gel in there. So you have something, we call it a comb. Is that right? Comb. Yeah. Okay. So you have wells in the gel, and then you prepare your samples, and usually you have a very diluted sample, and then you would, when you're ready, you would put your gel in a chamber that will run a current through it. And you put this, you have to remove, otherwise the current will not go through. And then you put it in, you add your running buffer, you make sure that all is filled to the line, and in here you have running buffer. Always use gloves, otherwise you have proteins from your fingers that will just migrate with the other proteins and mess up your gel. So, and then you take out the comb, and then you load your sample in each well. And then after you do that, you put this back on, you hook it up to your voltage source, and then a current will run through, and your protein, usually in the Lemley buffer you have a stain so that you can see that it's going down until you reach a certain point. You don't want the proteins to run off the gel and be in the buffer, so you want to stop it ahead of when the line is very close to the bottom but did not leak out. So you stop that, and then you take your gel out, and this nice compartment, which was not available 20 years ago, you can actually break the gel cassette, So I'm just going to remove this for now. So you put it here, you break it, like I said. And gently, you don't want to break your gel. Sometimes your gel is very fragile if you're using a very low concentration of acrylamide. So you break it and try to figure out on which side is your gel, because you can't see very well. So there, it's right here on this side. Hopefully it's not too sticky. Usually we use a wash bottle to squirt it out without breaking it. This is very interesting gel. I broke it. It's okay. You don't do that usually. You squirt it with water so it is not, like, very gently, and then you put it in the staining solution to stain the protein, which is a chromatography blue stain. So the proteins will stain blue, and then you'll see them later on the gel. Okay, I have a question for you. So you know that acrylamide is not good for you, right? And why am I touching it with no gloves? Because I already was exposed to it a lot, so I don't care anymore. No.
Is it because it's polymerized?
It's polymerized. So when it's polymerized, it's safe. When it's powder, it's not safe. When it's liquid solution, 40% liquid solution, it's not safe. When it's polymerized, it's fine. So this beautiful gel gets stained and then destained, because you don't want your gel, all of it, to be blue, and you won't see anything, and you use a destaining solution, and what remains is only your protein bands. So here's an example where we were looking at hydrolyzing soy protein, and we wanted to use enzymes that are specific to certain protein bands, to certain protein components, because proteins in soy is not only one single type of protein, we have different types of protein. So we wanted to target certain proteins to enhance particular functionality. So what we did is we ran a soy protein mix that did not have any hydrolysis, so that we see what are the components. We tried to run standards if we have standards, so to identify which is which. And then we ran one that has been subjected, in this case, to papain enzyme, and this one was subjected to pepsin enzyme. So what happened is we noticed that the papain enzyme targeted these bands, the beta-conglycinin. The pepsin enzyme targeted these bands, the glycinin subunits. So you can see from the gel what was the specificity of your enzyme. So this is one example. There are many examples where SDS pages is really useful, and this is just one example. Okay. Another fun electrophoresis is isoelectric focusing, which is the nightmare for Chelsea these days. It's been for months, not days, right?
Months. But we're getting there. We're almost there.
Okay, so to simplify things, you create a strip, also made a strip basically, that is, you created what we call pH gradient. So you have a specific electrophoresis strip, and you have ampholyte solution of ampholytes. Ampholytes are polymers that carry a lot of different charges. So there are different ampholytes that you can mix, and each ampholyte will have a particular pH because of the charges that it carries. And then when you allow current to run, then your charges will separate. Your ampholytes will separate based on their charges. The more positive will go towards the negative, and the more negative will go towards the positive, and then you end up having a gradient of different pH points on that strip. After you do that, then you load your sample onto the strip, your protein sample, a mixture of proteins. So and then you run the current. And the protein also is different charges, so the more positive will run towards the negative, and the more negative will run towards the positive. And then what happens once they reach the pH where they are at their isoelectric point, they will precipitate at that particular point and no more, move no more. They will stop there. So basically they reach their isoelectric point on that strip that has a pH gradient, and then they will stop. And then you separate them in this case based on their isoelectric point. So here's 2D electrophoresis. It's very common used in the world of proteins because of the fact that there are so many different protein components that you have in one mix, even from one source of a food. And then you want to see in this, let's say in case of Chelsea, she wants to look which proteins are more allergenic or react with antibodies and cause an allergic reaction than other proteins in a mixture. So separating the protein only on size does not separate them very well. So one dimension, because you might have multiple proteins with the same molecular weight, and to separate them only on the isoelectric point is not also enough, because some proteins might have the same isoelectric point. So if you do two-dimensional separation, then you'll end up having better separation, and you can better look at what mixture you have, and in Chelsea's case, for example, which one of these are more reactive than others. Okay, so in this example, what we normally have is we also have a cassette, same thing, but instead of having multiple wells, we get an acrylamide gel that has only one well, or one well for molecular weight marker, and the others is just one whole well, big well. So you run your first dimension, which is a strip of your isoelectric focusing, and you slide it in that one well space. And then now you have your isoelectric focusing strip with proteins separated on isoelectric point. And then you have acrylamide gel down here that will separate them based on size. When you run the current, they will all move down, because again, you mix them with STS or urea. In Chelsea's case, also urea is a denaturing solvent, reagent, and then they move down, all of them. Okay, so what you see here, you see this blue band, you had about 2 proteins that have the same isoelectric point, but they separated on the STS page part or the acrylamide part. They separated. This tells me that while they have the same isoelectric point and showed up as one band, they're actually 2 because they have 2 different molecular weights. In this example here, you have 2 very different proteins with 2 different isoelectric points, but if you run them on the gel that separates based on size, they tend to have the same size. So if we ran this without isoelectric focusing, these 2 bands will be 1 band. So this is a demonstration how 2D gel can separate even better your mixture. Okay. Capillary electrophoresis is also a cool tool for protein separation and uses a small amount of sample and is high throughput, very efficient for separation based on different modes of separation. So basically you have a capillary tube, a very small, thin, narrow diameter tube made up of fused silica, and also you have buffers, and you have 2 different electrodes that you run current through, and you have a detector on one side, and it's kind of like an HPLC detector, so you can have different types of detectors that you can hook up, and you will get separation in a capillary tube, and then you will get an absorbance reading or even an RI reading, or electrochemical detectors. So you can have multiple different types of detectors. The capillary tube can consist of different modes for separation. So it can be capillary zone. That means you have your silica that has silanol groups that are negatively charged. They pick up positive charges from the buffer that you're using. And then when the current runs, the charges will run to the other electrode and pull the different proteins along. And based on the charges of the protein, they will get separated. And then you can change the pH like you would do for ion exchange, and then you will get a better separation or enhance the separation. You can have it as a gel in there, so you get separation based on size. So if you have acrylamide, you can also separate the proteins like you did with the gel electrophoresis, but this will give you quantification. You will separate based on size and get quantification. You can also have separation based on isoelectric focusing, where you have that gel with the pH gradient. So different modes of separation, and the added advantage is you can have quantification. All right, let's take a moment, think about this one. Any thoughts? You have a thought, okay. C? C is a good thought, but any other thoughts?
A and C.
Okay, A and C. Well, nobody likes B?
All of the above.
Amanda settled it. It's all of the above. So, it doesn't matter if you start with an ion exchange then size exclusion if you want to do two-dimension separation, or if you start with size exclusion then ion exchange. It doesn't matter what the order is. It depends on the preference. So, obviously you have proteins that some of them has the same molecular weight but different charge, and some has the same charge but different molecular weight. So you can basically, you need two-dimensional separation. So separation based on charge and separation based on size. So A and C you can do either, doesn't matter. You can also use ammonium sulfate to precipitate the +2 charge. If you precipitate the +2 with a certain concentration of ammonium sulfate, you will have the +5 net charge in solution together. But they are very different in molecular weight. So one is 30 kilodalton and one is 90 kilodalton. So you can really use ultrafiltration. One of these membranes tubes that I showed you. So you can choose a 35 kilodalton cutoff. So your 30 kilodalton will pass through in the permeate, and then your retentate will have your 90 kilodalton, and you separated it. Well, this is very simple. You would wish that research is that simple, but usually it's more complicated. But it's just the concept that you can use any of these and get a separation. Okay. So there are a lot of characterization for proteins. We cannot go over all of them, but one important one is nutritional quality. Is a big one, especially for labeling when you want to report percent daily value. So you have to take into account the amino acid composition. You have to take into account the digestibility of the protein. So amino acid composition is very important because of the fact that you want to know if the protein is complete. Does it have all the essential amino acid, or is it limiting in some essential amino acids? And you want to know what is the first limiting amino acid. The first limiting amino acid meaning the essential amino acid present in the lowest concentration among the other essential amino acids. So let's say you have 9 essential amino acids. Which one is the most limiting? For example, in cereals, it's mostly lysine. In beans, it's methionine. And then I said digestibility. If you have high protein in your sample, but it's not digestible, or it's very digestibility is very low, then it's as if you're not eating protein, basically, because you cannot get the benefit out of it. And digestibility is impacted by what? Can you think of reasons why a protein might be less digestible than other proteins? What would make a protein non-digestible? Have you heard of a protein that is not digestible?
I know it's raw. Meat isn't as easily digestible as cooked because it hasn't been denatured. Well, denaturation of the protein is definitely important for digestibility, especially for globular protein. A lot of people eat raw meat in different countries of the world, and they're fine. But most importantly, vegetable source proteins are globular proteins. They're very compact, and if they're not denatured, the enzymes have a hard time reaching active sites or binding sites. But there are other cases where your protein digestibility is way, way reduced from original state. You're smiling. You're happy. Yeah. When the solubility what? What would make the solubility of the protein low? Other than that, you can have lipophilic protein and still is digestible. pH. Let's take pH out of the pH in the gut. You have pH 2, you have pH 7, you have pH 8. pH is not a problem. No. Are you going to tell me? You know this. Sarah? Bound to other compounds or bound to each other. So if you process the product in such a way that you've created huge polymers, large polymers, denatured and polymerized, oh, they're going to be huge, then the enzyme digestibility is going to be much reduced. So processing impacts protein digestibility. So you might start off with really good protein, but then process it some more, and then you wind up with low value protein.
How is digestibility affected by like coagulation or sulfur bridges or cross-linking?
Very good. So coagulation of cheese, for example. So you have, this coagulation is mostly ion source. It's not necessarily covalent linkages. So once you have that cheese, it's going to start melting and digesting by acid, solubilized by acid in your stomach, and then break out those polymers that form because of ionic bridges, for example. So that is not a problem, because cheese is not linked together via covalent linkages. Covalent linkages are the most destructive to digestibility.
Disulfide linkages?
Yeah, disulfide linkages, but that's not going to be in cheese or yogurt or regular food. It will be in really advanced heat processed food, will result in really large polymers of protein that are linked by disulfide linkages, covalent linkages, Maillard reaction based linkages. Okay, I was hoping to get through the lecture, but no, I will finish it on Friday and start vitamins on Friday too.